Welcome BNG426 526 class. Here is another flipped classroom pre-lecture. This is for Wednesday's flipped classroom assignment on metabolic pathway engineering, or at least conceptualizing metabolic pathway engineering. So we've talked about 5 main goals, let's say, for metabolic engineering. What do we want organisms to do? And one of them, and one of the key ones really, is to take an old product, scrap that, and have organisms make a new product. So we can use industrial organisms like E. coli, Saccharomyces cerevisiae, Ralstonia eutropha, etc. to make products, not only the products that they're accustomed to making, the products that are part and parcel of them surviving in the wild, let's say, but also novel products that we want them to make, that we know enough about the inner workings of the organism that we can fix them up and engineer them to make. It requires a in-depth knowledge of carbon metabolism. It requires us to know about the organism's carbon flux. In other words, where does the carbon go? A cell eats glucose. Where does that glucose go? We've talked obviously in depth about glycolysis and central metabolism, things that offshoot central metabolism like amino acid, biosynthesis, etc. And we've also talked a little bit, certainly one of the flipped classrooms was about a maybe not so typical carbon source, phenylacetic acid. How do organisms that are able to break that down, how do they do it? And where does the carbon go ultimately when they do it? And you saw with that flipped class that the carbon goes into, acetyl acetyl-CoA and succinyl-CoA. While acetyl-CoA could go to acetic acid, it also more readily would go to central metabolism, i.e. the Krebs cycle, the TCA cycle, whatever you want to call it. And so that's key, especially when we're thinking of phenylacetic acid as a xenobiotic, as something that needs to be degraded. So the goal for manufacturing, for biomanufacturing, for engineering these organisms is to redirect carbon flux from one product to another in this case. And that is also, as it turns out, the challenge. How do we redirect carbon flux from one product to another and make sure we get a reasonable amount of that other product, enough to be able to make a process out of, to make an industrial process? We need high productivity, we need high yields. How do we go from a product that cells make rather routinely, high yields, high productivity, to something completely different, something that cells are not used to making? And that's kind of the challenge, the main challenge. It's the goal and the challenge. So, first we start with the old, out with the old. What does an organism make that it spends a lot of its carbon on? And what can we do with that carbon afterwards if we don't want to make that product? So, for example, Ralstonia eutropha, obviously a bacterium I know well, and I hope you will know well by the end of this class. It makes this intracellular polymer called polyhydroxyalkanoate, and you can see that it just fills itself up. The cell fills itself up with a bunch of little, what are called granules of this polyhydroxyalkanoate. In fact, 80 to 90% of the cell's entire dry weight can be made up of this polyester, this polyhydroxyalkanoate biopolymer, bioplastic, whatever you want to call it. And it can be useful if you're interested in bioplastics. But say you want to try a new venture. You want to try maybe a fuel molecule or a small alcohol type molecule, which could also be a fuel molecule. How do we divert the carbon? How do we go from this cell full of plastic to a cell that's either full of isobutanol, which probably wouldn't work, but more likely a cell that would secrete out isobutanol that we could collect? So the simplest way to divert carbon from traditional product in microbial metabolism is to just eliminate the pathway or block the pathway. So block the pathway that makes polyhydroxyalkanoate, so it doesn't make it anymore. So you see cells under the microscope like this. They're not full of these granules anymore. They're full of just cell cytoplasm. And then with that, we say to ourselves, well, okay, now what is the cell using that carbon for? And that's where we come to in with the new. We want the cell to use the carbon to make that isobutanol. So when we knock out or when we eliminate the polyhydroxyalkanoate biosynthesis pathway, carbon flux is going to be upset in the cell. So the carbon is going to flow, but the flow is going to be upset to a certain degree, especially if we remove something that's like a carbon sink, like PHA. A carbon sink, it's stored. It's a storage molecule stored in the cells. This would be like if you're filling your pool, your swimming pool, with a hose and a spigot outside. You're filling it up, and then someone comes along and cuts out a 1 foot section of hose. So no longer is the water going to the sink, to the pool, but it's going somewhere else. And the key is, in the case of carbon metabolism, where is it going? In the case of the pool, it's just running into the grass. But here in polyhydroxyalkanoate land and Ralstonia eutropha, we need to find out where that carbon is going and how we can attach a new hose and repurpose that carbon for a different product. So as you all know, because I talk about it so much, Ralstonia eutropha produces naturally polyhydroxybutyrate, PHB, this molecule here, right here. And that is a well-known bioproduct, and we can modify it to a certain extent to make other slightly different polymers, but it's most well known for this PHB. So if we eliminate that ability of Ralstonia eutropha to make PHB inside the cell, to fill itself up with those granules, and it's something that's got to happen to all that carbon, because as you saw in the microscope image, that is a lot of carbon. So it's got to go somewhere. And what it does do, and where it does go, is that when you eliminate the ability of Ralstonia eutropha to make PHB, this results in a strain that secretes pyruvate, which is rather odd if you think about it. Pyruvate is a key central metabolite, but it's just being secreted. So why is that? How did this come to be? Why is the cell eliminating so much useful biomaterial here, so much of a key compound, a central metabolite? Again, it seems crazy. Pyruvate is pretty much the gateway to the TCA cycle. But then again, you've got to consider that at the time when Ralstonia eutropha is making this PHB, this polyhydroxybutyrate, it's not growing anymore. So maybe it's not using the TCA cycle as much. Maybe it's only doing amino acid recycling, so it doesn't need to gather TCA cycle intermediates to make different amino acids. There's a number of things that could be going on there. And the pyruvate pool just gets too big. So it's got to do something with it. And so what it does is it secretes it. Maybe in the hopes that other Ralstonia could use it. Who knows? But what it does do, and it has been shown, is that Ralstonia eutropha will secrete pyruvate if we eliminate the polyhydroxybutyrate biosynthetic pathway. Okay, so with a pathway eliminated, we want to harness that carbon flux. And I know carbon flux is something that I keep mentioning very briefly. And I keep promising that we're going to talk about it. And we will talk about it for sure. But we haven't really done that yet. So the key is not to think about carbon flux per se as a novel concept, but to think about it as, well, where is the carbon going in this case? And we'll get into flux analysis and all that later. But we can take advantage of this carbon imbalance that we've created by eliminating PHB. And the carbon that used to go to PHB can be channeled to a different product. we can pick that product. Let's pick isobutanol, for example. Then the question will be, or the question is, how to tie that new product, the making of that new product by the cells, to the cellular metabolism that's already there. And in this case, we're kind of left with a big clue here. Pyruvate is a central metabolite. We can start there. What can we turn pyruvate into? But first we got to think, where does pyruvate go in the cell? Like in a normal cell, where is it going to go? So it can come from a lot of different places, and it can go to a lot of different places. Anaerobically, it can go to lactate, it can go to acetyl-CoA. It could even go, if we do our engineering right, it can go to isobutanol, for example. So as a central metabolite, we can imagine this pyruvate going to lots of different places, and a lot of central metabolites do this. As we know, you've seen the pathway maps that I've shown in class. You may have read about a few of them in some of the papers that I've posted. And when constructing a recombinant organism, do we take into account the competing pathways, the things that compete for pyruvate? We could even call those things carbon sinks, because say we want to make isobutanol, we might theoretically, at least, want to channel pyruvate to isobutanol, and that's that. Unfortunately, with pyruvate being such a key metabolite, we can't shut everything else down. But how do we eliminate enough stuff, or just kind of hold back enough stuff so that we're getting something meaningful going from pyruvate to isobutanol? And that is kind of what we're going to talk about in the flipped classroom. But pyruvate isn't the only molecule, and you could even say isn't the only central metabolite with places to go. So ketoisovalerate, which is a key intermediate in making isobutanol, or would be a key intermediate in making isobutanol, also has plenty of places it could go. And some of those places are pretty important. You see a couple of amino acids on the list. So we've got some decisions to make. We obviously can't eliminate the synthesis of valine and leucine, but how do we see that enough carbon is channeled to isobutanol to make enough product isobutanol with which that we can design a process out of our Ralstonia eutropha strain that we want to engineer to make isobutanol? And so if all that is completely confusing to you, let me just simplify it a little bit. So again, we're thinking about taking metabolic pathways. From day one, we've thought about metabolic pathways. How to take them and basically plug them into different things. And the key is, do we plug them into different things completely and eliminate what they previously had done? And in some cases, you can do that. In this case, probably not, because you have amino acids to think about. So you have a variety of molecules that you could make, a bunch of intermediates that the cell can make, most of them naturally, and we have to add some enzymes. It's a fairly straightforward pathway to make alcohols, and these alcohols can be biofuel molecules. So basically we are constructing a strain that produces alcohol, or at least in principle, in practice, we're constructing a strain. On paper, we're doing this. Okay, so the overall goals for the flipped class exercise. First of all, again, let's keep familiarizing yourselves with what I call the lingo, the terminology that certainly I use probably more often than I should. The specific terminology for things like disciplines like molecular biology, the practice of strain engineering. What do I mean when I say heterologous genes? Well, heterologous genes are genes that are put into one organism that came from a different organism. That's essentially what heterologous means, right? We all know that by now. And what is a key intermediate? What is a key metabolite? What does that mean? Pyruvate's a key intermediate. What does that mean? Well, it means that the cell can't do without it. And in some cases, it might mean the pathway can't do without it. So there are a variety of things, but just, again, let's familiarize ourselves with the talk so we understand metabolic pathways more. And then, of course, metabolic engineering has its own lingo, carbon flux, things like that. So given what you know about pathway manipulations at this point, design a pathway, kind of like we designed a phenylacetic acid degradation pathway in E. coli. Design a pathway in a well-studied bacterial species. We're expanding the product range of the bacterium. So we were playing at the front end in our first flipped classroom exercise by making a strain that ate something else. And this time we're playing at the rear end, or the outputs, where we're looking at different products. It's making one product, let's get the strain to make another product. And expanding product range of the bacterium. Actually, what we're doing is swapping out products. So not necessarily expanding the range, but changing the range, the product range. And based on the provided papers that are going to be posted on my courses and the lecture that I'm giving right now, you'll work with your group to plan the construction of an organism that can take its central metabolites and make some biofuel molecule, make some alcohols. Okay, so things to do for this assignment. So first, watch this recording. Congratulations, you've done that. Browse the papers that I have posted on my courses for this assignment. Now, there's a lot of them. There's about 5 or 6. Definitely don't read them cover to cover. Just be familiar with what they're talking about. I reference some of them in the assignment sheet. And read that assignment sheet. And then come to class prepared to answer the questions, and answer the questions as a group. Talk them over. Again, I'll be roaming around like I usually am. Ask me questions. Ask me whatever question you want. And then just work it out. There's a few questions, as always. 8 or 9 questions. Work them out. Talk to them. Talk to your group about them. And then when class is over, hand in your individual assignment, and hopefully it'll be a complete assignment. And then that's that. Okay, so on that note, I will let you get to perusing those papers, and I look forward to working with you on Wednesday, the flipped class assignment.